Students, in last class we are discussing the white blood corpuscles. So here, based on the presence of the granules, based on presence of granules, the WBC is classified into two types. One we can call as the granulocytes, and second one is granulocytes. So here the granules are presenting. In granulocytes, the presence of granules in the cytoplasm of the WBC, presence of granules in cytoplasm of a WBC are called as a granulocytes. So here these granulocytes are classified into three types. These are classified into three types. It is classified into three types. One is three types. So such as I can call them as acidophils. Second one is the basophils, and the third one is the neutrophils. So here there are three types of the basophils, acidophils, and the neutrophils. Next, here absence of the granules. What are granulocytes means? The absence of granules in the cytoplasm of a WBC is known as an granulocytes. Here these are classified into two types. One we can call them as monocytes, and second one is called as the lymphocytes. So these are the four types, that is 5 types. So here the WBC which are presenting, we can call them as a polymorphonucleosides. Here WBC is also called as nucleosides. That means polymorpho means poly means many, morpho means externally, and nucleus. That means by seeing the nucleus, the sites mean cells. If the seeing the cells of the nucleus, we can easily identify which type of the WBC is presenting, so that we can call them as the polymorphonucleosides. Poly means many. Morpho means external. An external, and nucleo means nucleus. Nucleosides means the cells. So here the cells which consist of nucleus, by seeing the nucleus of these we can easily identify the type of a WBCs we can easily identify. So we can call them as polymorphonucleosides. So what are these A types? That are 5 types. We will discuss now one by one. We will discuss now. So coming to the first one, that is an acidophils. And the second one is the basophils. And the third one is the neutrophils. So here the acidophils. Here the acidophils in a red blood cell, that means white blood corpuscles, it appears 3 to 4% it is occupying in an acidophil, that is a granulocytes. These are the granulocytes. So here acidophils will occupies 2 to 3% in a WBC, in the cytoplasm of WBC. It occupies 2 to 3% it occupies, and also these are also called as acidophils, are also called as the eosinophils also. Eosinophils. Why they are called as eosinophils means they can be by using eosin dyes. They having eosin. By using eosin, we can easily identify the type of the acidophils by using the dye. That dye is called as eosin. With the help of eosin, it can easily identify, so that we can call them as eosinophils also. And here these will having the 2 to 3 lobed structures are presenting. Will having 2 lobed structures. These are 2 lobed structures. These are 2 lobed structures, and also these are added. That means these acidophils by using the acid dyes or eosin or acid dyes. By using acid dyes, here it becomes that is here these are becomes in red in color. That means when using dyes, it becomes a red in a color. These are produced a red in a color, and morely these are presenting in an allergic conditions. Allergic conditions. That means here they secrete, they produce more amount in an allergic conditions. They are morely producing, so that we can call them as they are called as acidophils, or we can call them as the eosinophils. Next, coming to the second one, that is the basophils. Here basophils will occupies 0.5 to 1%. These are presenting in white blood corpuscles, where here these eosinophils are having an S-shaped nucleus. An S-shaped nucleus is presenting. S-shaped nucleus is presenting, where that is in basophils, and these are having, that means these are as having the basic dyes. By using basic dyes, we can easily identify the by using basic dyes, we easily identify the basophils that are will produce a deep blue color, are produced deep blue color is produced. So due to the deep blue color, we can easily identify the basophils by using the basic dyes. And also here these basophils are mainly helpful in inflammation. They are mainly helpful in an inflammation. So here they produce 3 substances. It produces 3 substances. These are produces that are, one we can call as a heparin. Second one is called as histamine. And third one is called as the serotonin. So these are the 3 substances which are secreted by the basophils. So here heparin, it is used as an anticoagulant substance. It is used as an anticoagulate substance. And here histamine, it acts as the vasodilators. Vasodilators. And serotonin will act as the vasoconstrictors. That means vasodilators means it helpful for increasing the lumen of the blood vessels. We can call them as the vasodilators. So here vasoconstrictor means it helpful for decreasing the lumen of the blood vessels. So they are called as the serotonin. That means if any inflammations, that means inflammations are taken place, here they increase the lumen of blood vessels. So they are called as vasodilators. If they are not presenting, any inflammations are not presenting, here they constrict the blood vessels. So we can call them as the vasoconstrictors. These are the three substances which are produced by the basophils, where these are helpful in an inflammation in a structure, where when they are used basic dyes, it produces a deep blue color is produces. So such type we can call them as the basic basophils. Next, coming to the neutrophils. Here neutrophils will occupies it occupies the 60 to 65% of the WBC. 60 to 65% of WBC is occupied by the neutrophils, where here they use these are having 2 to 3 lobed structures. These are having 2 to 3 lobed structures are presenting, where here will having 2 to 3 lobed structures are presenting, that is structure, and they use a neutral dyes. Here they using a neutral dyes. Here due to the producing of a neutral dyes, it produces the purple color. It produces not a purple, it produces the pink color. It produces a pink color producing. So with the help of these pink color, by using neutral dyes, we can usually obtain their pink colors are producing. So that here these neutrophils which are presenting, these are mainly helpful in these are mainly helpful in the that is, we can call that as neutrophils are mainly helpful in the phagocytosis. In the phagocytosis means they engulf the bacterial cells and round up them and kill them. So such type of substances we can call them as the phagocytosis. And also the neutrophils which are presenting, we can call them as the microscopic policemen also. These are also called as the microscopic. These are also called as the microscopic policemen. Why means here they engulf the bacterial cells. They engulf the bacterial cells and helpful for killing of by the process of phagocytosis. They kill them. So such type of process which is taken place by the neutrophils, we can call them as that means they engulf. Here engulf the bacterial cells, engulf the bacteria and round up them and kill them. So such type of process we can call them as the phagocytosis. So that here neutrophils we can call them as the microscopic policemen, where here it produces neutral dye. By using neutral dyes, it produces a pink color or produces where having 2 to 3 lobed structures are presenting. So these are 3 types of the granulocytes, where having the granules in the cytoplasm of the the cytoplasm of the WBC will having the granules are presenting. Due to the presence of the granules, we can call them as they are called them as a granulocytes. Now coming to the second one, that is So now coming to the second one, A granulocytes. Granulocytes. So in this A granulocytes, here in the WBC, in the cytoplasm of the WBC, the granules are absenting. So due to this, we can call them as A granulocytes. So here A granulocytes are of two types are there. One we can call them as monocytes, and second one we can call them as the lymphocytes. We can call them as the lymphocytes. So here the monocytes and the lymphocytes. So here these monocytes as well as the lymphocytes which are presenting these monocytes and lymphocytes, we can call them as A granulocytes. Why means here the granules are in the cytoplasm of WBC, the granules are absenting, so we can call them as A granulocytes. So here a monocytes will occupies here the monocytes will occupies the 2 to that is will occupies 20 to 30%. 20 to 30% will occupies the monocytes, where here will having the monocytes. So here coming to this monocytes, already I said 20 to 30%, not 20 to 30%. Here will occupies a 3 to 5% will occupies the monocytes in a WBC, where will having here will having the monocytes will consist of a kidney shaped nucleus is presenting. A kidney shaped nucleus is presenting. So by using dyes, here they produce a blue color is producing. Here they produce a blue color are producing, where it occupies 3 to 5% and having a kidney shaped nucleus is presenting. And by using dyes, which color it produces means here it produce a blue color it is producing. And also here monocytes we can call them as monocytes. We can call them as macroscopic. They are called as macroscopic policemen. That means neutrophils we can call as microscopic policemen, and the monocytes we can call them as the macroscopic policemen, where these are also will helpful in the process of the phagocytosis. That means they engulf the bacterial cells and fight against the harmful germs they are producing. So these are also helpful in the process of phagocytosis. These are presenting. So here we can call monocytes are called as a macroscopic policemen, where they having when they using the dyes it produce a blue color is produces. So such type are called as the monocytes. And coming to the last one, the lymphocytes. Here lymphocytes will occupy the 20 to 30%. 20 to 30% will occupy the lymphocytes in a WBC, where here will having the lymphocytes will having the large nucleus, will having a large nucleus. Large nucleus is presenting in the cytoplasm of the WBC, will having a large nucleus is presenting. And also here by using dyes it produces a pale blue color is produces. It produces a pale blue color produces where these lymphocytes are presenting in the lymph nodes. That means the new lymphocytic organisms they produce. These are present in lymph nodes as well as lymphatic organs. These are presenting where these lymphocytes will produce the antibodies. It produces the antibodies where these antibodies which are presenting they fight against the harmful germs. Here these lymphocytes which are presenting will be differentiated into two types. It is differentiated into two types. One is called as the B lymphocytes, and the second one is the T lymphocytes. That means here the lymphocytes are classified into two types. One we can call as B lymphocytes, and second one is called as the T lymphocytes. B lymphocytes means the lymphocytes which are produced by the bone marrow, they are called as B lymphocytes, and the lymphocytes which are produced by the thymus gland. If they produce the thymus gland is producing, so such type of lymphocytes are called as the T lymphocytes. These T lymphocytes and B lymphocytes here they produce an antibodies are produced. So when these antibodies which are producing that due to the vaccination also these vaccines, that means due to the vaccinations these antibodies are also can be able to produce. They live for longer duration and which produce antibodies and fight against the harmful germs where they are stored in the form of lymph nodes and lymphatic organs. These are stored, and whenever required they produce the antibodies are produces. And also here we know that here these we can call them, that means WBC we can call as a polymorpho nucleosides. So here we'll having already we discussed, that means here acidophils will having two lobes and basophils will having the basophils are having S-shaped nucleus, and neutrophils having 3 to 4 lobes lobes are presenting, and in monocytes will having a kidney shaped nucleus is presenting, and in lymphocytes will having a large nucleus is presenting. So by seeing the nucleus of the 5 types, that is acidophils, basophils, neutrophils, monocytes and lymphocytes, by seeing the nucleus of these 5 types we can easily identify which type of that is WBC is presenting. So with the help of these we can easily identify, by staining the nucleus we can easily identify the type of the white blood corpuscles. So that here white blood corpuscles are called as a polymorphonucleosides. Poly means many, morpho means externally, nucleo means nucleus, sites means cells. By seeing the cells of the nucleus, here by seeing the external nucleus of the cells we can easily identify the type of WBC, that are granulocytes or agranulocytes. So these are the that are the 5 types which we are discussing in WBCs. Now coming to the functions of the white blood corpuscles. The functions of a WBC. Here the functions of a WBCs, what are that function means? First one, it acts as the phagocytic cells, phagocytosis, and second one, it takes place an inflammation, and third one, they produce antibodies. It produce and antibodies are produced. So here phagocytosis, here already we know that the phagocytosis means they engulf, that means they engulf the bacterial cells, engulf bacteria and round them and kill them, engulf and kill them and take as food particles. So such type of process we can call them as the phagocytosis. We can take in place where here the monocytes and the neutrophils are helpful for the phagocytosis in conditions. That means here these are helpful in the monocytes and the lymphocytes. Next, inflammation. Inflammation is mainly taken place by the basophils. Here these are mainly taken place by the basophils, where here it becomes the redness, the swelling, the redness of the wound, and also the swelling of the wound, where here it helpful for the producing, where these are enters, these basophils will enters across the capillaries. They enters into the across the capillaries, across capillaries, where they are entered across the capillaries, called as diapedices. Diapedices. We can call this as the diapedices. That means what is diapedices? Means if the WBCs are oozes outside across the capillaries, then such type of process we can call as the diapedices. So this will be taken place and helpful for killing of the wound, the germs. Where when these are produced kills, here they produce a pus. That means an yellow colored substance are produced that we can call as a pus, where that is the WBCs are fight against the harmful germs are produced. That will taken place by the second process called as an inflammation, where redness of the wound and swelling and reduces the rate of the swelling, and also redness and fight against the harmful germs. So such process we can call as an inflammation. And the last one is the producing of an antibodies. These antibodies are produced by the lymphocytes. These are produced by the lymphocytes, where these lymphocytes which produce antibodies, these during, that means when germs are weakened, here what happens due to the vaccination, due to the vaccination, here it produced the more amount of germs are produced and helpful for killing of the body whenever required. That means here during vaccinations, the germs which are enters into our body, whenever they enters, here automatically our lymphocytes, immune system will respond, where they stimulates the lymphocytes, and lymphocytes will produce the antibodies and helpful for killing of the harmful germs which are enters into our body. So these are the three functions which is performed by the WBC, that is phagocytic nature, inflammation, as well as producing of an antibodies. Now coming to the third one. That are blood platelets. The blood platelets, or otherwise we can call them as the thrombocytes. So here the blood platelets are thrombo means clot. Thrombo means the clot. Thrombo means the clot. Sites means cells. That means the cells which are helpful for the clotting of the blood. That means the cells which are helpful for the clotting of the blood, then such type of cells are called as the thrombocytes, are also called as the blood platelets. These blood platelets are they are oval or round shaped in structure. They are oval or a round shaped structure. Round shaped structure where these are produced from the megakaryocytes. These are produced from megakaryocytes of the blood. These are produces where these are very numerous. That means these are will producing. These are having 2 lakhs to the 4 lakh. The cells are present for cubic millimeters are presenting. This means 2 lakhs to the 3 lakhs of the cubic cells are presenting in the blood platelets in the blood. These are presenting 2 lakhs to the 4 lakhs of cells are presenting for cubic millimeters. These are presenting and if you see in the lifespan of these, here the lifespan is the lifespan of the blood platelets. The lifespan of blood platelets. The lifespan of the blood platelets is 4 to 5 days. Here the lifespan of the blood platelets is 4 to 5 days only. And also here the producing of blood platelets. That means producing of blood platelets is called as platelets is called as thrombopoiesis. Thrombopoiesis. What is thrombopoiesis? Means the producing of the blood platelets from the bone marrow we can call as the thrombopoiesis. And also the increasing the number of, that means increasing the number of blood platelets is called as thrombocythemia. That means thrombocythemia means increase the number of blood platelets. When these increase the number of blood platelets means if any cuts, when it is produced means the cuts, here the cuts or injuries are taken place. If injuries are taken place, here the increase the number of blood platelets are taken place. So increasing the number of blood platelets we can call as is called as a thrombocythemia condition. And the number of decrease, here the decreasing, here we can write here decreasing the number of the blood platelets is called as the thrombopenia. So here what is thrombopenia means? Here what is thrombopenia means? Decreasing the number of, here we can write this is the decreasing the number of, we can call as the thrombopenia. We can call that as the thrombopenia. That means if number of individuals, that means the number of the blood platelets are decreased, then we can call as thrombopenia. And if it is increased, the number of blood platelets are increased, we can call as thrombocythemia. Conditions are taken place. And also here there is a condition, that means these are the blood platelets are mainly helpful in the clotting of the blood clotting. So here the blood clotting, if any injuries or cuts are taken place, here main function of the blood platelets means if any injuries or cuts are taken place, here within a fraction of seconds the clotting will be taken place. That is mainly due to the presence of the blood platelets. So such process is called as a blood clotting. This for the blood clotting, here that is consist of an enzyme called as a thrombokinase enzyme. Thrombokinase enzyme. So in the presence of thrombokinase enzyme, here it helpful for the clotting of the blood will be taken place. So along with the thrombokinase enzyme, here calcium ions are also an important, where these thrombokinase enzyme will produce the prothrombin. It produces a thrombin. This thrombin is secreted by the liver, where it is produced by the liver, where this prothrombin in the presence of thrombokinase, it converts into an active thrombin. It converts into an active thrombin. Along with this, vitamin K is also helpful for this. It also having a vitamin K is also helpful for the clotting of the blood, that is produced by the thrombokinase enzyme. It is produced. Vitamin K is also helpful for the clotting of the blood, that is will be helpful for the clotting of the blood. How this will be taken place, we will discuss now. Now coming to the mechanism of the blood clotting. The mechanism of blood clotting. The mechanism of the blood clotting. So here the blood clotting that is taken place, here will consist of the prothrombin. So here the prothrombin is produced by the liver, where these are present, these are produced, these are present in the plasma of the blood. It is produced the plasma of the blood, where here it consists of that is a prothrombin, and also contains the thrombokinase enzyme. It is an activating enzyme. Activating an enzyme. It is an activating enzyme, and also here it consists of the calcium ions. Here the calcium ions. Here the clotting of the blood. Calcium ions. Here without calcium ions. Without calcium ions, clotting will not be taken place. Clotting not be taken place. And also here will consist of a vitamin K also will required, and a vitamin K, which is a fat soluble vitamin that is consist, which helpful for the clotting of the blood. And also here will consist of the fibrinogen, which is an unstable compound. It is an unstable compound which is produced by which is present in the plasma of a blood, where this is unstable, and where it is dissociates into a fibrin threads, which are these are unstable or insoluble. These are soluble and these are insoluble. These are stable, insoluble fibrin threads. So here how this mechanism is taken place means, here they produce the prothrombin. Here they produce the prothrombin. prothrombin by the enzyme called as the thrombokinase. Thrombokinase, here it produces an active thrombin. Here inactive, it is inactive, and here it is an active, where here calcium ions are also presenting. So that thrombin will act upon the fibrinogen. Thrombin will act upon the fibrinogen. It acts upon the fibrinogen, where this it acts upon the fibrinogen also. Here this fibrinogen and converts into an active fibrin, that is stable, that is soluble. These are non-soluble. These are soluble and these are insoluble. So here by acting upon the calcium ions along with the thrombokinase, so here this thrombokinase will act upon the fibrinogen and converts fibrin. Here this fibrin will produce an insoluble fibrin threads. Fibrin threads. The producer, that means these will produce the fibrinogen, will produce the insoluble, the fibrin, insoluble fibrin threads. These threads will forms into a mesh-like structure, which helpful for the clotting of the blood. That means here these fibrin threads which are forming, these fibrin threads will form a mesh-like structure. So that mesh-like, when they forms a mesh-like structure, the cells, bacteria, that means the blood cells are deposited in these fibrin threads, so which are insoluble, where is it helpful for an easily clotting of the blood, where it forms a mesh-like structure forming. These mesh-like structure, fibrin threads which are forming threads in the form of mesh or network-like structure, and helpful for deposition of the blood cells in this. So it helpful for easily a clotting of the blood will be taken place. This is how the clotting of blood is taken place in organisms. Here the main role of thrombocytes or blood platelets are helpful for the clotting of the blood within a fraction of seconds. If a clotting of the clotting mechanism will not be taken place in a disease called as a hemophilia. Here hemo disease. Here you are having a disease that is a hemophilia. So here in this hemophilia, small cuts are taken place, or from the gums of the, that means from the teeth of the gums, if the blood uses, here cannot helpful for clotting of the blood, where small cut is also helpful for the hemorrhage condition. Hemorrhage means continuously, here it leads to the hemorrhage. Hemorrhage means the continuously the oozing of the blood. Continuously the oozing of the blood is taken place. It is not helpful for the delay of clotting is taken place, where it leads to the death of an organism. Such condition we can call as a hemorrhage, a condition that we can see in a hemophilia, which is a genetically disorder, that is a disorder, that is sex linked and inheritance is taken place, where this character already we studied the sex hormone, that is sex linked character, sex linked inheritance. So in this sex linked inheritance, here X linked inheritance, where here that is carries from carrier daughter to the from the father to the carrier daughter, from carrier daughter to the grandson. These characters are presenting. So here it shows that a crisscross, an inheritance are taken place, or zigzag, an inheritance is taken place due to the hemophilia, where it is a genetical disorder, where it leads small cuts will helpful for the death of the organism due to the hemorrhage in conditions. So this is how, that means prothrombin, from prothrombin to thrombin. Thrombin is converted a fibrinogen into a fibrin active, a fibrin insoluble, that is insoluble threads. It is a fibrinogen is soluble where it is present in plasma, and that is converted into active by the thrombin. It is converted into an active thrombin in the presence of calcium ions. So that here in the help of thrombin, it converts the fibrin threads, where it helpful for the carrying of, this is helpful for the formation of the blood clotting, where in the form of a mesh-like or network-like cells are deposited and helpful for the clotting of the blood. This is there it takes place the mechanism of the mechanism of blood clotting.